Good morning, everyone. Welcome to another session of Outside In, which are a set of ecology talks which are curated for students, and we target these at older school students, but everyone is welcome to attend these. So this week we have Dr. Deepa Agashe, and I'd like to welcome her. And Professor Sadamni is going to talk about a little bit of Deepa's work. So, Professor Sadamni, over to you. Thank you.
Thank you. Thanks to the communications team for ably organizing this and tremendous efforts. And I would like to thank Deepa for kindly agreeing to give this talk today. Deepa is trained in ecology for a number of years, and she had joined NCBS. So we all know that DNA is nothing but a string of nucleotides, and proteins contain a number of amino acids. It is known when the protein synthesis happens in the cell that 3 nucleotides stand for 1 amino acid, and this goes on. But the third position, the third nucleotide, is usually considered as very flexible, even though certain amino acids are alone recognized, the tRNA is recognized. So the third amino acid, third nucleotide being flexible is viewed as what we call as a wobble hypothesis. But then what Deepa had shown using bacterial genomes is that even in the change in the third nucleotide matters in the protein expression, and it could affect the growth of the organism and so on. This finding questions the wobble hypothesis. So that's very interesting work by Deepa, and she has also worked on insect nutrition and behavior. Deepa was elected as a vice president of the American Society of Naturalists, and she was also been awarded the SERB Women Excellence Award this year. In the webinar today, she plans to talk about how bacteria coexist and human bacteria coexist together harmoniously mostly, but not so harmoniously at times. With this, Deepa, please.
Thank you. Thank you very much, Sadamini, for coming up with this wonderful idea of having this seminar series. I sure hope that I can, tell you, get my own excitement about the fascinating world of symbioses across to all of you. Thanks also to the comms team for organizing and being here for support. Okay, so I'm going to get going now and share my screen. I have a few videos and slides to share with you. So give me a minute. One second. All right, there we go. Can everybody see my screen?
It's visible, Deepa.
Okay, great. All right, so let me get going then. So I'm going to talk to you today. Whoa, what's going on? I am having a bit of trouble getting my Q&A box to be visible despite my screen sharing working. Okay, I'm just gonna close that right now because I don't think it's gonna help.
Yeah, Deepa, at this point, if there are any questions, we'll just alert you to them. Don't worry.
Yeah, that sounds great. Yeah, sorry. Okay, gotta turn this off. Okay, all right. Apologies for the glitches. So I'm going to tell you today about incredible partnerships in biology, and to sort of extra bonus points to anybody who can guess what this image is showing here. But don't worry if you can't guess, I'll come back to it later in the talk. And so I really want to focus today on symbioses or partnerships in biology that as one of the partners, because for many reasons they are in my mind really extra fascinating compared to any partnership, right? So we all have friends, we all have various kinds of partnerships that we enter during our lifetimes with friends, with spouses, with children, with parents. And all of these sort of, in some sense or shape or form, encompass the core ideas or the core concepts that I will discuss today. How do you keep partnerships going? How do you form them? How are they broken? How do you maintain them? What's the currency? What's the trade that's happening in a partnership? So we'll touch upon some of these questions, which are really fascinating. And one of the reasons I think microbes are particularly fascinating as one of these partners is because the difference between these microbial and eukaryotic or animal and plant partnerships are really, really uh, enormous, right? The differences between these organisms, that's something. The numbers are also different. So microbial partners are typically vastly outnumbering whoever else they're partnering with. And so that adds another element of interest. The microbial partners typically reproduce much faster as well. So they have the potential to evolve faster at a faster rate compared to the other species who are hosting them very often. And so there are all these different aspects to this sort of trade game, which make it really, really fascinating, much more so than when the partners are sort of more equal or balanced in some sense, right? So let's dive in then. And I think some of you are at least thinking about, well, what's the big deal? Why are we discussing symbioses again? We know already a lot about symbioses, right? Those of you who are in school or have been in school recently, you remember your textbook, which says there are these classical examples of symbioses, and we know so much about them. But I want to sort of highlight today that there's actually a lot even about these textbook examples that we don't know a lot about yet. And we are still discovering lots of new things about these very, very interesting partnerships that we supposedly know a lot about. And so let me take one example to begin with, which is this example of lichens, which many of you have heard about or have definitely seen around when you go outside on a walk on the barks of trees, especially if it's not rained recently. You see these crusty lichens, right, very often. And they look like moss sometimes, and they are very, very diverse species. And lichens are fascinating. As I said, a textbook example of a relationship between a fungus and an alga. And that's kind of what we've learned so far. And it's really fascinating to think about lichens because they were the reason German scientists in the 1800s had to coin the word symbiosis, because this was the first time that they realized that two organisms are living together. And over time, the sort of literal meaning of this word, which is just to live together, has come to also mean or imply a mutualistic benefit, right? So both partners are gaining something out of this relationship. And so symbiosis now is something much more than just cohabiting. It also has connotations of being beneficial for the partners, much more than that. So what do we know about symbiosis, and what about lichens? So I want to start off by showing you this video, and let's see if that works out.
So most people think a lichen is a moss. That's kind of where it ends. To the untrained eye, it could just look like another branch or another leaf or another twig, or even a piece of bird poop or a piece of gum on a rock. They don't understand them. If you don't know that it's alive and it's an organism, you might gloss over that. These things are often scale. You realize the actual microtopography of that landscape is actually huge. Lichens are a mini ecosystem. It's not one organism. My name's Tim Wheeler. I am a lab manager at the University of Montana. But kind of my passion is fungi, specifically lichens. By trade, I'm not a photographer. I consider myself a naturalist. I took a forestry class with a professor, and he pointed out these lichens growing on the trees, and he was like, hey, we really don't know anything about these things. And that kind of piqued my interest. So I just kind of casually started collecting them. The 10th or 12th lichen that I collected, I didn't recognize it. I thought it was cool and new, so I sent it off, and it turned out to be a new species to science. That doesn't happen if you study birds, and I kind of got hooked. Historically, a lichen was considered part algae and part fungi. As we've dug into these things deeper, we're finding that it's more complex than just these two partners. There's other fungi in there, there's other algae, cyanobacteria, other bacteria, all working together, basically in this little mini ecosystem. The fungus produces the structure, right? So think of it as like a little greenhouse, which inside that they're growing algae. The algae are photosynthesizing, the fungus provides water and other nutrients, minerals and stuff like that. The diversity of lichens is huge. We have foliose, which are your big leafy lichens. They have an upper and lower surface. You have these fruticose lichens, and these are your 3D or branching lichens. And then you have the crust lichens, which are kind of crusty, right? If you can just draw a crazy shape in your head, in your mind, that probably exists in the lichen world. That's what I like about lichens. You have these things that are flat and boring. You have these things that are incredibly long-lived and incredibly branching. Any complex 3D structure you can probably imagine, you can probably find an example mimicking that. It is exciting when you find that perfect specimen. It has all the characters. It's pristine condition. You want to be able someone to look at it from a human perspective, but you also want to be able to show those smaller details all in one shot, right? And so my photography skills grew out of the need to illustrate these specimens, these species. The collection started because you needed to come back to kind of verify your IDs. Back here I have, I think, just over 16,000 specimens, so that's 17 years of collecting. I have kind of one wall that I've been trying to contain it to. I don't think I'm ever going to stop collecting lichens, so my numbers are only going to go up. It's kind of like a checklist, kind of like birders have a life list. You feel good that you've captured that organism. So once you might only have one little plane that can be in focus. So in order to overcome that shallow depth of field, my camera is attached to a rail, and every 125 microns it'll take a picture. That gets sent to the computer, this program, he'll come focus, and they stack them, and they take every little sharp bit from every layer and make it into one continuous sharp image from front to back. And the result is one picture. When I first started stacking pictures, I would everything was in sharp, nothing was out of focus. And because I thought that's what was important. But as I got into it more, I kind of shifted more towards the art. A little bit of depth, a little bit out of focus in the background actually makes the rest of that stuff pop a little more. Sometimes I'll wet it if it needs to be rehydrated. You might have this boring little gray lichen on a rock, but when it gets wet, it could kind of unfold and kind of almost flower and turn into this brilliant blue-green thing. It's always exciting to see them come alive. You can almost take the specimen out of its habitat, kind of raise it up to this glorified color, shape, everything can be perfect. I think a lot of scientists, they have the little point and shoot camera. For me, that just didn't cut it. I needed to make them, the center of attention. I don't think the general public knows the importance of them. They're notoriously sensitive to air pollution, to changing temperatures. And so if you have a changing climate, what's the first thing that's going to respond are these fungi. You can kind of get a trend of how your climate is changing because these lichens respond so quickly.
Science Friday. Thanks for watching. If you liked this video, then you'll love our other science documentaries. Be sure to subscribe to our channel, then join our growing community of supporters on Patreon.
Okay, great. So that's enjoyable for you. I really like this video. So I want to build upon what this person was saying in the video, right? About these lichens. We initially thought they were fungi and algae, and in the last few years we've discovered a lot more. There are other partners that play a role. It's not just a bipartite. It's not a two-person or two-partner relationship. So here's an example of something really fascinating that was only reported 2 years ago, or discovered 2 years ago. On the left, you see this picture of Bryoria. There are two kinds of Bryoria. This is a lichen found in North America, and the Native Americans there knew that the yellow one at the bottom is toxic and to avoid it, whereas the brown one can be used for various things. And when researchers started looking at, well, what's the difference between these two, they found that the yellow one and the brown one pretty much are very similar to each other. There doesn't seem a big difference when you sequence their genome, that is, look at their entire DNA. And so they were sort of trying to figure out what is going on here. And what they discovered is a third partner which plays a role. perhaps in this relationship. And so if they took one of the thallus, which is one of these strands that you see in the lichen, and they cut it, they chopped it, and they looked at the cross section of this, which is what you see on the left image here, and you look at and you paint the different partners in different colors, you find that there are actually 3 of them, right? So the basidiomycete here, which is the new fungus that they discovered playing a part somewhere in here, it's colored in green. The purple is showing you the traditional partner that we know of for a long time. And the pink is the alga, which is again something that we know for a long time. And you can see that all 3 of them are forming very nice, interesting layers within the cortex of this lichen, and they're all there. And what was even more fascinating is they did a survey of many different lichens which were similar to this lichen, so this group of lichens across the world, and they find that most of the species that they investigated seem to have this fungus, this basidiomycete fungus, which is not known to be parts of lichen before. And so there's fascinating new things to sort out here. What we don't know yet is what exactly this new fungus, this basidiomycete, does. Is it producing something? Is it producing the toxin? And the researchers suspect that without this fungus, the lichen would not be able to function the way it does function right now. What seems to be happening is the yellow version of the lichen seems to have a higher proportion of this new fungus, basidiomycete. So maybe it's producing something that is important for making this yellow one toxic, right? So there's more research that needs to be done. Going on. One example of how we think we know something, but we actually don't, and there's a lot more fascinating stuff to be discovered. And again, last year, something else was overturned. Our thinking about corals and sea anemones as again being sort of 2 partner organisms. We know now that there's a 3rd partner in sea anemone, again found in many different corals that were examined by these researchers in many parts of the world. And they've called it corallicolid, right? This 3rd partner. So we know already that there's an anthozoan, which is the animal part of the coral, which is providing the housing structure. The dinoflagellate is the microbial symbiont, which is doing photosynthesis, providing energy for the symbiosis. And then these 3rd corallicolids are interesting. The 3rd guys who are now being found in these corals and sea anemones, they belong to a group of species called apicomplexans. And apicomplexans are a large group of organisms, animals that include malarial parasites, right? So many apicomplexans are parasites. That means that they're not really symbionts in the sense that they don't provide any benefit to their host. They're just sucking stuff off of the host. Now, what's interesting is in the corallicolids that they examined, which hang out in these corals, they're also embedded inside the coral, okay? So they're also intracellular, which means they're inside the cell. And these corallicolids, they share features of both parasitic and non-parasitic apicomplexans, right? So they seem to be intermediate between the very many parasites that we know of, which are apicomplexan, but they're not quite completely parasitic. And what's also fascinating is that corallicolids have the genes that are necessary to synthesize, to make chlorophyll, right? So they could photosynthesize perhaps, except they don't have the downstream machinery that's required. They can't make the rest of the proteins that are essential to actually finish photosynthesis, right? So they're capable of making chlorophyll. And this by itself should be actually pretty toxic to them, because the making of chlorophyll involves a lot of oxygen radicals, which should be toxic to the cells. And we really don't know how they deal with it. And again, much more remains to be discovered. So this is fascinating. This is another example of a symbiosis that we thought we understood, but turns out we really don't. And it seems to be widespread. Okay, so now I'm sure that many of you are thinking, well, how do the partners find each other and how do they stay together, right? It's all very well to say, okay, fine, there's a symbiosis and it's cool and it's great, but how does it begin, right? And once it's established, let's say you are a host for a symbiont, how do you make sure that your future generations are going to get the right symbiont? And so there are different kinds of answers. In some cases, the mother can help. So the mother can give you the right symbiont that you want if you're a host. If that fails, then you can find your own perhaps from the large set of microbes that we're all floating in effectively, right? So as I'm sure you've heard, there are all kinds of fungi and viruses and bacteria that are present in soil, in water, in the air. They're present on our skin, inside our bodies. And so we're kind of floating around in a mess of microbes. And it becomes a very important question then to ask or to understand, well, how do partnerships work? How do these partners find each other and how do they stay together, right? So here's a schematic from a review paper that people published recently that sort of gives you a broad dichotomy of how these things work. Like I said before, there can be direct host-to-host transmission, and many times this involves passing on the right bacterial or fungal endosymbionts or ectosymbionts to the offspring, to the babies. And for example, in cows or ruminants, in termites, in humans, and honeybees, also in some beetles and bugs, the mother will often pass on the right symbiont to the babies, right? So here's a picture on the right of a protaspid bug. And this is the mother bug who cares for the offspring, cares for her babies for a long time. And you see beneath her a mass of eggs, right? And maybe you can see there are two kinds of eggs there, some that are red and some that are white. The red ones are the ones that are actually going to become little baby bugs, but the white ones are the ones that are actually what's called trophic eggs, okay? And these trophic eggs are only meant to be nutritious sort of kind of eggs that are not fertilized, right? So when the babies hatch out red eggs, they will go and consume and they will eat the white eggs. What the mother does is she puts this white mucus on top of all of these at the right point before the eggs are going to hatch. She also makes sure that the eggs all hatch at the same time, right? So that everybody gets a chance to get this mucus. And scientists have shown that this mucus contains the symbiont or the bacteria, which is critical for these bugs to survive and do well, right? So this is really, really interesting. That's one example of how this mother to baby transmission of symbionts works. In other species, this doesn't seem to happen. The mother doesn't really pass it on, but instead each host has to get its symbionts from its environment, okay? And it has to then put in a filter that says, okay, well, if I'm just eating, for example, a lot of different microbes through my food, I'm going to get all kinds of species, and many of them I don't want inside, right? I don't want because they're not beneficial. They're not useful to me. So I want to have a filter in place. And so many animals like mosquitoes or flies or worms or some fish seem to have these filters in place where they can actively regulate which microbes make it into their guts and live there and thrive and produce something that they want in exchange, and which microbes do not get a chance to do that, right? And then there's a very interesting third category for which we have very few examples, relatively speaking. And these are also an interesting category. This is caterpillars are shown here, butterfly caterpillars as an example, where they don't seem to actually have any bacteria inside their guts. And this is unusual in the animal kingdom so far, right? That most animals which people have examined seem to have microbes in their guts, and they can help them in breaking down polysaccharides, so complex sugars which we eat. They can provide new nutrients. So in many animals, the bacteria essential vitamins or minerals which we don't get from our food. They can also help defend from pathogens. They can also help us acquire food. So there are lots of insects, for example, which feed on plant sap, which has tons of stuff missing in it in terms of basic nutrients, right? And so if you don't have these bacteria in your gut that process whatever is in the wild plant sap and turn it into something that the host wants, these insects would not be able to survive in these environments at all. In some cases, the microbes help detoxify, right? So you're eating something really toxic, and without the microbes, you cannot eat it, right? So there's all these different benefits, and caterpillars seem to be an interesting exception where they don't seem to really care about the microbes in their gut. They have some microbes in their gut, but they seem to be just sort of coming and going depending on whatever the caterpillar is eating. So this is a really example as well. And we in the lab, I'm not going to talk too much about it today, but I just want to mention that in my lab, we've been working in the last few years to understand some of these interesting systems and symbioses. And so we worked with 3 different insect systems. We work with butterflies and dragonflies, and we sort of captured them from around campus and in different parts of India. And we asked whether they host microbes in their guts and whether these microbes are important for these insects. And we find, by and large, to our surprise, that butterflies and dragonflies seem to host a bunch of microbes, but they change depending on what the insect has eaten. And depending on the geography, the insect may be eating slightly different things. And so that seems to change the microbe, what's called the microbiome, or the collection of microbes living inside its gut. And so we think when we did experiments with butterflies, we were able to remove the bacteria using antibiotics. And we find that the caterpillars are fine even when we feed them antibiotics, right? So this sort of corroborates this idea that has come up in the last few years, that there are actually some animals which don't seem to have strong symbioses, at least when it comes to food symbioses with bacteria. And that's something that we're investigating now. Other people are investigating as well, trying to figure out how does that work? How can they survive without microbes? Because everybody else seems to really need microbes and depend on microbes. And then we also work with a beetle called flower beetles. They're everywhere, they're a pest species, and they are associated strongly with humans for many, many years now. And we are actually glad that they are a system where they do seem to depend upon the microbes that they find in their environment. And they kind of fall in between in the spectrum of host-to-host transmission and host filtering, because they live in flour, right? So they're living in this box of flour, looking out everywhere, and all the life stages of this beetle are living in the flour. They're eating the flour, they're pooping in the flour. And so what this means is any bacteria that are in the gut of these beetles, or any fungi or any other microbes, are going to keep getting recycled, right, through the flour. And so in a weird way, it is both host-to-host transmission as well as through the environment, because the environment is so deeply, like they're so embedded in their environment that this dichotomy kind of slightly breaks down for these beetles. And so we are now investigating. We know already that if we remove the microbes, again, by giving them antibiotics, the beetles don't live as long, they don't grow as fast, they don't survive as well, but they're not essential for the beetles. So if we get rid of the microbes, the beetles suffer a little bit, but not too much. They can still manage. And so we're now trying to understand what is the role of these microbes for the beetles, which microbes are important, which bacteria are critical for these beetles, and right, what is the currency that's being exchanged here for this trade. So this is work that we're doing in the lab, and happy to talk more about later if you wish, but I'm not going to focus on this at the moment. I'm going to talk about work that other people have done, which I find really, really cool. Okay, so the next question that I hope some of you are thinking about is, well, who makes all the decisions in this partnership, right? Is it one partner or is it both partners? And they sort of, how do they decide? How do they negotiate this? And so the answer is it seems like it's more of a constant tug of war, that it's not one partner who's always in the commanding role and who's dictating terms of the partnership, but both sort of are constantly trying to get the upper hand and get a better deal for themselves. And so what do we know about this? Again, I'll use a couple of examples to talk about this. Here's a really, really interesting example of plant root and symbiont fungi. So this is the picture I showed you before at the beginning of the talk. Here is cells of plant root. So if you squint, you can really see the details of the root itself. And in the green color are marked these symbiont fungi. So these are called arbuscular mycorrhizal fungi. And these are very, very widespread in soils. And what's really fascinating about these fungi is that they form these long structures with hyphae, right? And these hyphae allow a single unit, a single organism, to be able to attach itself to many different, or associate with many different plants and plant roots, okay? So there's this whole network in the soil, underneath the soil, that is composed of these fungi. And conversely, a single plant may associate with many different mycorrhizal fungi at the same time. And we know after many years of work, scientists have figured out that one of the key things that they exchange is phosphorus, okay? So plants have a time accessing phosphorus, which is a nutrient that's important to all life forms. And the fungi do a better job of taking up the phosphorus, and then they can give it to the plant root, okay? And then in exchange, the plant is thought to give them carbon, okay, which the plant is able to acquire because it photosynthesizes, right? So that's the exchange that's happening. And so these scientists wanted to figure out, well, who's calling the shots here, right? Like, does the fungus, because in space, if you think of a field, an agricultural field where different amounts of fertilizer are put in, or there's different kinds of soil, and some soil may have more phosphorus, some may have less phosphorus. And you have these mycorrhizal fungi that are sort of spread out across these big patches. So do they decide which plant and which plant root gets phosphorus, and how do they do this, right? So to do these experiments, they did this really nice work where which is what you see here in this diagram. They took host roots, right? And they allowed the roots to be spread across these two different sections towards the bottom of the plate. And in both sets, they put fungi, which the hyphae that you see here, which then connect with the host root. The fungi are either marked with red color or with a different blue color, right? And they invented this sort of method of tagging the phosphorus. Sorry, the fungus is not marked, it's the phosphorus which they supply to the fungi are marked with two different colors. And then they can watch, so you see here the red tagged and the cyan or blue tagged phosphorus. And then they can watch how this phosphorus is transported to the host root in the third section of the petri dish. And whether it's the red, which is put in more, or the blue. And then what they do is they put in either more red tagged phosphorus or more blue tagged phosphorus, right? So they're creating an imbalance in the fungal network. One gets more phosphorus and the other part gets less phosphorus. And they track it using this fluorescence microscopy. And they figured out that if you give more on one side, the fungal network redistributes to the host root. Okay, so they managed to redistribute the phosphorus to make everything more or less equal across the network. So the fungi are playing a really interesting role here, where they're taking phosphorus where it is in excess and transporting it to where it is less in amount, and allowing the host, or different kinds of hosts perhaps, to be able to sample more phosphorus. And the speculation is, by doing this, the fungi increase their chances of getting more and more carbon from the host, because unless the host has phosphorus, it doesn't survive very well and can't give them carbon in exchange. And again, this requires more work. This is more recent stuff, just came out last year. And we're still learning really, really interesting details of how these relationships are modulated and how they work. I want to go on to another example, which I'm sure many of you have heard about already. So this is the Hawaiian bobtail squid, which has a fascinating symbiosis with bacteria called Vibrio fischeri, which they filter in from their environment. Okay, so they're living in seawater, and they filter the Vibrio, and they use it for an interesting camouflage strategy. So I'd like you to see this clip.
It's hard to hide in the ocean, but the Hawaiian bobtail squid is a master of disguise.
During the day, if they're disturbed from the sand, they'll come out and just sit on the surface with a sand coat on them, trying to be invisible. And if that doesn't work, they'll eject out ink and leave that blob of ink behind in their shape.
Their camouflage strategy at night is even more amazing. Luminous bacteria actually live inside the squid's body and light up its underside, helping it hide from.
So they're emitting ventral light. They're camouflaged against the moon or the starlight so that they don't cast a shadow. And so as not casting a shadow, it's like a Klingon cloaking device.
With support from the National Science Foundation, biologist Margaret McFall-Nye studies the squid's remarkable light organ, the small pouch that's home to the cloaking bacteria Vibrio fischeri. Its structure has a lot of similarities to an eye.
We can look at all parts of the light organ, so all parts of where the bacteria go into the light organ and then reside.
To better understand how the bacteria take up residence inside the squid, McFall-Nye's team studies the babies under the microscope.
These protrusions right here, which we call appendages, help to filter, basically bring in bacteria from the seawater. So, you kind of have this little cave, this kind of nest.
Sees the squid condos and the nursery.
I mate the squid every 2 weeks. A female will generally lay eggs about every 3 to 5 days. This clutch is a fairly good size, probably 200 eggs or so.
Analyzing this squid bacteria symbiosis could help develop new camouflage materials for the military or lead to new pharmaceuticals to fight bacterial infections.
At the very basic level, we're asking how animal cells talk to bacterial cells.
A conversation worth listening in on. For Science Nation, I'm Miles O'Brien.
Okay, so I want to quickly show you a bit more detail of this light organ that they just mentioned in the video. And this is part of the light organ, and this is where the bacteria come in, and they're marked here in green. And you see that they sort of get inside and these sort of take up residence. Now, what's really fascinating in terms of control that we were talking about, who controls this relationship, they figured out that that these squids sort of go through cycles where every day in the evening, which is when they really use this light camouflage based on bioluminescence coming from these bacteria for camouflage, they filter in more of the bacteria in the evening, right, before the night comes and before they need these bacteria. And then the bacteria proliferate and grow inside the light organ, and they produce this light, which the squid uses. And in the morning, when it doesn't need it anymore, it chucks them out, the squid, right? So there it seems like the squid is really controlling how many bacteria make it in and which bacteria manage to come in. So that's a very different aspect of control. And so again, it seems like there are different components to this tug of war, and sometimes somebody is in control, and maybe after solution, the other partner may be in control, and this keeps going on. So it happens after a long time, right? So some of these relationships that I've been talking about have been around for millions of years. How do they sustain and do they sustain for such a long time? And in short, very interesting things happen, right? So here are some examples of what happens. So I'm going to make a cartoon of sort of a lot of work that people have done on this. And if you sort of vaguely, broadly think of the time spent in a partnership, that means evolutionary time. So we're talking millions of years time scale. And you start with bacteria that are free living on the left here, that become sort of facultative over time. That means the relationship is not essential, what I mentioned for the beetles earlier. It's good to have the bacteria around, the symbiont around, but it's not essential for the host. Like the host can manage without them. And then we think many times it eventually relationship becomes obligate. That means that neither of the partner can really survive without the other, right? So here's what happens. So each of these circles represents, it's a cartoon image of a bacterial genome, right? So there are maybe 3 different bacteria here which are associating with different hosts. And let's say their genomes are like this to sort of normally begin with. This is what free living bacterial genomes may look like. There's some variation in size, but they're sort of large. Now, over time, what happens is as the relationship becomes facultative, these genomes start shrinking. They start losing genes and they start becoming smaller. And they lose genes, we think, partly because of just chance events that happen. So mutations happen all the time. And through a process, what's called genetic drift, these bacteria may effectively lose genes that are no longer under strong selection, which means they don't affect their survival because they're living in a nice, comfortable environment. Just give them food, making sure that it gets transmitted from generation to generation. And the bacterium can afford, in some sense, to make more mistakes and lose some of these genes, which it would not be able to afford if it were living outside on its own, right? So you have this bacteria that lose their genome, it starts shrinking. And by the time you get to obligate relationships, so bacteria which are now sort of wedded to this host, these genomes are really tiny, right? So if you think of some of the smallest genomes that we know in bacterial species across the world, they tend to be belonging to these obligate mutual relationships with some host or the other. And what's really interesting is now this can be taken to the extreme, right? where in some cases, these genomes become so tiny that they're kind of useless for the organism, for the host. In some cases, like in the case of the cicada, Magicicada, which belongs to a group of cicadas called Magicicada. They have these 13 and 17-year life cycles in temperate areas. These genomes have even fragmented, right? And they've become even smaller and smaller and smaller circles, which are, and many more of them. And that effectively prevents the cicadas, prevents cicadas from being able to cross with each other. And the endosymbionts can no longer, they're split into multiple different lineages, right? And so this is really, really fascinating. So this is where a relationship can go for so long and has all these repercussions that are very difficult to then come back from, because now once you've lost all these genes, it's very hard to gain them again. Okay, here's another example of such a relationship and what happens when it goes on for a long time. This is the citrus mealybug. Maybe some of you have seen mealybugs like this on plants in your garden or outside. They're kind of white and fluffy and small, and they're also sticky if you poke them. They release juices, and they also have these endosymbionts inside their bodies. These bacteria which they're housing. So this particular mealybug has a really fascinating, interesting relationship where it's got not one but two different endosymbionts. Okay, and what's really interesting here is they both of them are living one inside the other. So the mealybug cell nuclei in this picture are shown in blue. So that's, remember, the eukaryotic cell is much bigger. So this is the mealybug cell. And the blue is marking the middle, more or less, of the cell, which is where the DNA of the mealybug is there. Then the Tremblaya bacteria are one of the symbionts, which are marked in red, and they're sort of inside that cell. And the Moronella bacteria is a second kind of bacterium, which is living inside the red bacteria. Okay, so it's like a Russian doll system where there's one bacterium which is living inside another bacterium, and this bigger bacterium is living inside the cells of the mealybug, right? So you have this. And what's interesting is, like I said before, the average bacterial genomes shown by these circles are this big, then the endosymbiont genomes, which you see below in red and green, tend to be smaller, right? And they may gain a little bit, perhaps some genes once in a while. They may exchange genes with each other, but they tend to be smaller. And so what the mealybug has done is it's taken up in its own genome some genes from old bacteria that it used to associate with and added them into its own genome. And those genes can now compensate for the genes that are lost from these new symbionts that are in the process of shrinking their genome, right? So that the mealybug is still sort of managing to make sure that it has everything it needs from its symbionts, except the old symbionts are gone, but it's maintained its genes inside them. So that's a very interesting trick that the mealybug is playing here. And here's another way to think of the same relationship. And I want to go a little bit more detailed to show an example of, what is this kind of action. So here's a cartoon again. And what you see in the cartoon is these outer cell is the bacteriocyte, which is this a special tissue where the host sort of harbors these different bacteria. And the yellow is the Tremblaya, which was red in the previous picture. And the green little blobs inside is the Moronella, right? Now, what people figured out recently is that at the interface of the cell wall of this Moronella bacterium, which is the green one inside, it has to make its cell wall, right? How does it do it? So what this host has done, interestingly, or maybe what the bacterium has done, is it shipped some of its genes to the host genome, okay? So these are marked by this star here. And like I said before, the bacterium has to make its cell wall or some components of its cell wall. And how does it do that? And look at this very, very fascinating strategy. So what happens is the first few enzymes are the ones that this bacterium to the host genome, okay? They're made by the host in the host cell and then shipped back into this Tremblaya and Moronella system, okay? So then these sort of enzymes and components come together and they form this chain reaction where some products are produced. Then those products are taken up, and inside the bacterial system, they've now acquired genes from other bacteria, okay? Maybe while they were free living, maybe while they were inside the host, that's not quite clear yet. And they've taken up these different genes, and now they use these different genes and proteins produced from these different genes to form another part of this chain of reactions. Then they take some of those and they finally use their own genes and their machinery to then make this component called peptidoglycan, which is basically a sugar that has to go inside and makes the cell wall, right? So this is a very fascinating example of how this whole system has now evolved to be really complicated, where all are doing very different things to produce one final end goal. That's really fascinating. And there are other examples of this as well in other symbioses that we know of so far. So now what happens if your partner disappears? Like, all of this is great. You reach this nice complex system where you really depend on each other. You can't really do without. What if one partner goes away? Then what do you do? So maybe you find a new symbiont as a host, right? And maybe, and this is very, very fascinating work, you convince a parasite to be good to you, right? And let me give you some details. So here is a species of cicada. Okay, so this is a cicada that you see on the left side here. I don't know if you've seen cicadas before, but they're the ones that go, that are singing in the summer, right? When it's very hot and dry outside, where they go king, 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 right? Maybe you've heard it. And so on top in the cicada, so you see its eyes and the bottom. And a cicada which has been dissected, right? And you see what's the ovaries of this cicada, this female cicada, are marked by ovi. And the special tissues which the cicada makes to house bacteria, the endosymbiont, are called bacteriomes, marked by BC. And these BCs are, the bacteriomes are shown in a more sort of zoomed in shot in the last picture here, right? So you have these bacteriomes, which are, as you can see in this middle image, are quite a large mass, right? So they're a very big investment by the host into housing bacteria. And these bacteriomes, if you squish them and you stain them with markers for bacterial DNA, you see that chock full of bacteria, okay? So effectively the cicada over evolutionary time has evolved to have these bacteriomes be basically sacks full of the bacterium that it needs to provide it with some important amino acids and other nutrients, right? So here, this is what is known already in many cicadas in the world, particularly in the US, where a lot of this has happened before. The cicadas have two bacteria typically, okay? One is called Hodgkinia and one is called Salcia. And both these bacteria are sort of housed in different cicadas or sometimes in the same cicada, and they're producing different aspects of what nutrition the cicada requires, okay? Now, this is work by a Japanese team, and what they figured out is that that in some of the cicadas in Japan, they don't find one of these bacterial partners, okay? So this is a situation that I was describing earlier, that you've lost a partner. So let me walk you through these really beautiful images. On top, you see the top row here is showing you one particular species where you see both bacterial symbionts are present, the pink and the green, okay? And that's marking DNA from the bacteria. You see that the bacteriomes have both of them, as well as okay? And the fat bodies in insects are where they store fat, and they're used, they're very important for females to be able to lay eggs and so on. And on the very right side, you see images where you see under a microscope, you're able to see sort of more detail of what's going on here, okay? So on the second row, you see a situation where you find that the host DNA is there. One of the bacterial symbionts, the pink one, is there, but the green one is missing, okay? And that's Hodgkinia, which doesn't seem to be there. Instead, what you find is marked in yellow, which is a new fungal symbiont, and they're calling it yeast-like symbiont, or YLS here. And that seems to be present in the fat bodies as well, okay? And similarly, in the last row here, there's another species of cicada, which again doesn't have the Hodgkinia or the green ones, has the pink bacteria, but also has the yellow fungus, okay? And so they figured out that this fungus is now of a symbiont and has some of these genes perhaps which are missing or because the Hodgkinia symbiont is somehow lost, right? And so they trace this series of events, evolutionary events, on a phylogeny. And so if you haven't seen a phylogeny before, it's simply a way to show evolutionary relationships between organisms. So if you see this tree-like thing here, the tips of the branches of this tree are showing you current species that they find. These are cicada species. And every time two branches collapse into a single node as you go backwards in time, which is towards the left of the screen, that is an event where there's a shared common ancestor, Okay, so if you go very, very far back in the tree, you will eventually get to the common ancestor of all cicadas that exist in the world right now, right? Okay, and so now on this tree of cicadas, some of these species are shown here in the pictures on the right. What they did is they said, okay, they did some analyses to figure out point of time in this evolutionary tree was the Hodgkinia, the green bacterial symbiont, lost? And at what point was the yellow yeast symbiont gained? Okay, and what you see here, that's shown with triangles in the branches of this tree. What you find is, interestingly, every time the loss of Hodgkinia occurred, so green triangle is there, you have a gain of a yeast-like symbiont, which is the yellow triangle, right? So see, 3 times in different parts of this, so different groups of cicadas, it seems like again and again the same thing has happened. They've lost an important bacterial symbiont, and around the same time they've gained a new yeast-like symbiont, right? And they did some work then finally to figure out, well, what is this symbiont? You know, what is this yeast-like thing? So it's a fungus, so yeast are fungi. But what does this fungus do? And it's very surprising what other fungi that are related to this fungus or this yeast are normally doing in nature. So I want to show you another video. Okay. So this is an ant that's walking along in a forest, and you'll see what happens to this ant. So this is, okay. Wasn't sure if what I would say would be audible while the video was going on. But effectively, in the video, you saw that this fungus called cordyceps or Ophiocordyceps, which is a very well-known parasite of many insects and other arthropods. This fungus is closely related to the fungus that they found to be a symbiont in these cicadas, right? And there are also a few other cases where, similarly, insects or hosts have co-opted a parasite which very often infects them and twisted its arm into basically being a good symbiont and providing it with some benefit because it needs it, having lost its old partner, right? So there are very interesting dynamics that are going on in this symbiosis world where, you know, relationships change. What was a parasite may become a friend now, and maybe that relationship also changes eventually, right? So we're still learning about these kinds of things. And so where are we? Just quickly closing, where are we with symbiosis? I would say what we know is fascinating, and I've just given you a small glimpse of it. There are lots more discoveries that are waiting, and I think the future is very exciting. So I'll stop here and I'll take questions. I'm sorry I could not figure out how to take questions in the middle of the talk. I would have really liked to do that, but I'll stop sharing at the moment, and maybe we can go on to questions now.
So we have one person who's raised their hand. We'll let Raghavendra Gupta talk right now.
Okay.
Raghavendra, please go ahead. Okay, I guess they don't want to ask the question right now.
Okay.
So let's just take one from the Q&A. So Satyanarayana asks, how does the mother find the right symbionts for the babies? And does this happen in humans as well? How is this phenomenon working?
So what sometimes happens is simply that the mother is full of these symbionts, right? So her own body does have the symbionts inside, and so what she does is when she's laying the eggs, when she's or producing the babies, she basically smears the babies with this symbiont in some shape or form, or she lays eggs which are extra eggs for the symbiont, for the babies, which can then be eaten to be able to obtain the symbiont, right? So these different kinds of strategies work, and they really depend on the mother having the right symbiont already in her body. As to how, if there are multiple symbionts in the body or other bacteria that are pathogens or parasites, how does the mother make sure those do not make it to the eggs? I don't know the answer to that, and I'm not sure that people know very clearly how this works for many systems either. But I think in most cases, when there's one very obligate, strong relationship, it seems like everybody else is already sort of filtered out in the mother's body, so that's a relatively easy step to do.
Great. So there's a question about whether there's any evidence for intelligence in these organisms being dependent on their gut microbes.
I don't know that there's work on intelligence, and there are problems with even quantifying intelligence to begin with. So I think it's a bit of a hard question. But I do think what's interesting, people have shown in mice that if you don't allow the mice, the fetuses, as well as the baby mice to have access to their normal gut flora, their brains do not develop like normal, right? So there seems to be some merit in saying that this organism's normal brain development is not going to work out as well without access to the regular microbes. So I think there's that. But in terms of sort of quantifying intelligence, like I said, there's problems with that to begin with. So as far as I know, we don't have any such evidence for that.
Okay. Well, I guess we have a lot more to learn. So Raghavendra has still got his hand raised, so I'm just gonna try again. Otherwise, we'll move on to someone else.
Okay.
Okay, never mind. So we'll go to Harshit Mishra. Harshit, please ask your question.
Hello, am I audible?
Yes, go ahead.
Good morning, Deepa ma'am.
Yes.
Very big fan of your work, firstly. So I have three questions. The first is, actually, I've always been thinking about this symbiotic associations and relating it to phylogenetics and stuff. So can you give me another system, like you spoke on cicadas and the microbial associations, squid microbial associations. So and you also said you worked on dragonflies and butterflies, and you saw that there was no such dependence on microbes. So I am interested in invertebrate biogeography and phylogenetics. But now through this webinar, I'm also interested in all these lichens and fungi and bacterial associations in insects. So can you just help me out with another model system which we can study in India? Like, I'm a bachelor student for my master's PhD.
Exactly what you want to study, but in general, I mean, look around you. There are so many insects at the very least that you could study. There are cicadas, there are aphids. Both of these systems are very well studied in other continents in terms of their symbionts and in terms of their own phylogenies. And in India as well, it's not impossible to do this work. So I would strongly suggest you to take up one of these systems. You could also work with plant systems, which there's a lot of really exciting work happening in India on plants, and they also form symbioses with different microbes. So I mean, I guess you have I would say you have the opposite problem of, there's too much choice. You're spoiled for choice.
Okay. And ma'am, like you showed that fungi growing out of ants and spiders, I've also seen a documentary where there are mind-controlling fungi in snails.
Mm-hmm.
So do we have these model systems even in India, like this fungi growing out of insects and...
Well, so I have for a similar looking fungus. I haven't sort of, I haven't gotten it identified to ask if it's the same or similar species, but I would imagine so. So I'm sure that they're there. And unfortunately, we don't have enough background or baseline work done in India already, but there's no reason you can't be the first to do it. So
yeah.
Go ahead.
Thank you, ma'am.
Sure.
So Sakshi Sani has raised their hand, so we'll allow Sakshi to ask their question.
Good morning, ma'am. This is Sakshi.
Hi.
Ma'am, I have a question regarding bird behavior and how microbes are affecting bird behavior. We have studied the examples where the human's behavior is affected, or the human physiology is affected by the microbes, but have we studied any examples in the birds?
In birds, actually, as far as I know, and I am not entirely sure about my understanding, but as far as I know, in birds, there are not very many good examples of microbes affecting, having a large impact on them, at least when it comes to gut microbes and so on. So I don't think so. For parasites, yes. So we know quite a lot about parasites in birds, and they do definitely, influence bird behavior and evolution and their ecology. But in terms of sort of symbionts that live inside their guts or inside their cells, I don't think there are very good examples of there being a very strong relationship which affects evolution in the ways that I have described today for many insects and plants. So yeah, I don't know of any examples. I don't think there are very good examples that are known.
Thank you, ma'am.
I think we can go back to our Q&A box. We have a lot of questions there. So one question is, how does the host filter bacteria from the environment, and is there any specific mechanism they employ? And how are they so specific?
Yeah, so the mechanisms are actually, where people have studied it, they're quite different in some cases. So in some cases, what happens is, for example, in an insect, in a bug, a particular kind of insect, there's a particular constriction between different parts of the gut. And that constriction, that barrier, can only be passed by the symbiont, which is able to move and which can swim. In some cases, the symbionts are the only ones that are able to tolerate a very acidic pH or a very basic pH in the gut. So there are pockets where only the symbiont can live and nobody else can live. So there's that kind of filtering mechanism. And in some cases, the host releases specific nutrients and so the symbiont then gets a boost, right? So the host, because this symbiont is the only one that can use these nutrients in some sense. So there are very, I guess there are a wide range of different mechanisms that can be used by hosts to filter out specific symbionts. There are also other mechanisms in the sense that you can enrich your surroundings for the specific symbiont, perhaps that you want to have. But I don't know of very many good examples of this. So I guess there are other ways that are possible, but the ones that we know of are largely physiological, where in the gut of the host, there is a way to either physically or chemically filter out non-symbiotic bacteria from entering or establishing in the gut. Yeah, so that's that. I see a couple more questions based on plants. Pavitra?
Yeah, go ahead with that question on the lichens on areca and rubber palms and things like that.
Right. So the question is, we've observed the lichens on areca and rubber palms, and these trees have become weak over time. Don't the plants exhibit defense mechanism against lichens? I'm actually not the best person to answer this, because I'm not sure if the lichens actually harm the plant. I would be surprised if that is the case, but I don't know enough. So I'm sorry, I'll have to look this up. If you could email me later, I can try and find the answer and let you know. Okay.
So we also have a question on plant roots and fungi there, which are releasing many secondary metabolites. So how do you use them to trigger the immunity so that the plants can resist infection? Again, this is a plant-based question.
Right. So the question is, fungi and plant roots release many secondary metabolites. Honestly, I don't know enough about this either. I'm sorry, I'm not really a plant expert, and so I can look this up again and maybe get back to you if you could email me.
So how about turning parasites into friendly symbiotes? How does that happen?
We don't know the mechanisms very, very well in most cases. In some cases, it seems the host is able to produce chemicals or other compounds that sort of prevent the parasite's parasitic machinery from activating. And in the absence of that, if it's able to suppress the parasite, then the parasite could eventually be useful to the host as a symbiont. But the details of the kinds of examples that I showed you today are not actually very well understood yet. So with the Ophiocordyceps fungus, it's not very clear yet. How does the host, how have these host cicadas managed to tame this parasite, right, and prevent it from being an opportunistic pathogen? I guess the simple speculation would be that they provide them with so much benefit that the parasite any parasitic mutants that don't become parasites and sort of kill the host are favored. And so if the benefit of cooperation is much higher, then the cost is not worth paying of being a parasite. So I would imagine that is how things work out, but the details are going to vary, I would expect, across different systems. So the precise, extra candy that the host provides to its parasite to convince it to not sort of be a parasite, I would expect would change depending on the system and depending on the organisms involved. So I don't think there would be a very general answer to this beyond what I just
So we have kind of an interesting. Sorry.
Yeah, go ahead.
Yeah. So, so someone is asking, is there a specific limit or a maximum number on symbiosis, or is there anything that you are aware of, where it has the maximum number of symbiotic organisms?
That is an interesting question, and I don't know the answer at all. I know that there are species of hosts that seem to have symbiosis with many different taxa at once. Humans seem to have symbiotic relationships with multiple bacteria that live in our guts. But I don't know that there is, that what is the largest number known to us, and is there like a theoretical limit to this number? Good question. I'll have to think about that a bit.
Yeah, and again, someone is asking, can one organism be symbiotic with one species and parasitic with another?
Absolutely. Yeah. So there are some very nice examples of this where, somebody who's a parasite actually is parasitic through, a mutualistic relationship. So in some sense, if you consider lichens that are living, sort of occupying real estate, right, on trees, or you think of endophytes, which live on plants. They're kind of parasitic sometimes, but the only reason they survive is they have their own symbionts, which help them do the job of being a parasite. So yes, that absolutely does happen, and that's fascinating as well.
Great. So we're gonna let some people ask their questions live again. Shivana, please go ahead. Shivana, please unmute yourself and ask your question.
Yeah. Hello, ma'am.
Hello.
Ma'am, I'm from GKVK. A very excited lecture indeed. And I have two questions. And the first question is, See, usually the insect will transmit the diseases as a vector, right? And will these pathogen means the vector, the pathogen will be turned to be as an symbiont? Is there in such kind of cases?
Not that I know of, no. Typically what happens is, well, in some cases, the vector is just a passive vector where it's not itself affected by the parasite. But in other cases, the parasite completes some part of its life cycle inside the vector. So it's actually parasitic on the vector as well. Yes, I don't know of.
Yeah, why I ask this question is because there are some bacteria. You said that there are some symbionts which they reduce their genomic size. Probably like that could be one of the like exciting factor where these vectors, I mean the microbes, they may reduce the like the genetic mechanism and they'll become as a symbiont to the insect. So that was a reason behind this question. And second question is, once the particular symbiont function is over, once the particular particular microbe is over, will the insect keep such kind of microbes in the future? Because at the end of the day, it's an investment.
So there's actually very interesting examples of control that the host has, right? So I told you already of the squid example where the squid, throws out the Vibrio during the daytime when it doesn't need it, and then takes up new Vibrio cells and in the evening acquires the light. Similarly, there's a very fascinating example of another insect, a bug, where actually a beetle. And this weevil, what it does is it takes up, it has these bacteria which it houses in a special pouch, right, next to its gut. And what it requires these bacteria for is to produce amino acid precursors. And those are important for the beetle to harden its elytra. Okay, so its back is hard and brown. And that cannot happen if the bacterium is not there. So what the beetle does is once it is ready, the adult has come out and eclosed and its back is nice and brown and dark and hardened, it gets rid of the symbiont, right? So it can recycle the symbiont. What happens is the beetle's own cells will engulf the symbiont and mash it up and destroy it and eat it. And only a few different symbionts will go and attach themselves to the ovary of the beetle, right? And once they're in the ovary of the beetle, then they be passed on to the next generation. But they are not they're not sort of housed and maintained inside the gut or the pouch of the beetle for very long. So once the job is done, the beetle gets rid of them.
Thank you, ma'am. Thank you.
Maybe we can look at a couple of the questions from YouTube. So Shivashish Indra asks, why do some organisms need a symbiont to survive and others can live without them? I think this is something you are working on. So, and what's the evolutionary significance? So
that's a very great question, and we don't actually know the answer yet, right? So this is something that we're still trying very hard to figure out. Other groups in the world are also trying to figure this out. Why are some guys very dependent on symbiosis? Some can sort of be okay with it sometimes. If it's there, fine. If the symbiont is not there, it's also fine. And some very way, and we don't honestly know the answer. We suspect a number of different factors that could play a role. One factor might be, how reliably can you find your symbiont? How often can you hang on to them? Is the symbiont going to be available whenever you need it? Is it going to be able to give you the benefits that you require? Are there other easier ways in which you can get the same benefits, right, without having a symbiont? Can you, for example, produce your own vitamins and minerals or acquire them from your diet? Then you may not require a symbiont, perhaps. And so it seems like the possibilities are many, and we don't have one real answer for this question. But it's absolutely a fun question to think about, and there's a lot of work that we need to do to follow up.
So maybe we can take just a couple more questions. So there's a question about, how can we sort of harness these symbiotic relationships for human health, if that's possible, such as using tapeworms, is there a way to build symbiotic relationships?
I don't know about building symbiotic relationships. That seems to be a bit of a tough problem. But I think what we can do, I mean, people are certainly working on this, scientists are certainly working on this. What we know for humans is there's a lot of individual variation in the microbiome. Okay, so what lives, the microbes that live in our bodies are at different parts of our bodies. So, you know, any two of us, if we figure out what microbes we host, they're very different from each other. Well, not super different. We clearly have similar looking microbes as a species compared to, for example, if you compare with any other species. But there's a lot of variation, and people are trying to use this fact to understand you know, what set of microbes in what composition and what portions are good for me may not necessarily be good for the next person, you know, to maintain maximum or top health. And so we know, of course, of specific sets of bacteria that are able to, in our gut, which are able to compete better with toxic bacteria like Clostridium difficile, which causes a lot of stomach disorders for people. And so we can use those specific interactions between different bacteria that all reside in our gut to be able to sort of outcompete one bacterium which is not good for us, using the other symbionts in our guts themselves. So those kinds of strategies people are certainly trying to use, and they do have some success. What we can do, of course, is if we figure out what is the symbiosis dependent on, so what chemicals it's dependent on, so these beneficial bacteria in our gut, what are they producing that makes them beneficial to us? If we can we could also maybe chemically synthesize those compounds and then use them directly instead of relying on the symbiotic bacteria themselves, perhaps. There are problems with this strategy, but it could at least work in the short term. And so that is another aspect of research that people are working on for sure. So I think building a symbiosis from scratch is a bit hard because you're trying to manipulate another organism into doing something that, you know, you expect a system to work in a particular way, and that doesn't always work out. So what we know from evolution experiments in the lab or in the wild so far is very often the evolutionary path that ends up being taken by systems is very different from what, you know, we would imagine or we would expect. And so there's a lot of chance and unpredictability built into evolution in nature. And that is what I think makes these kinds of systems very challenging. To be able to build your own symbiont is not straightforward or easy. Because evolution takes paths which we cannot predict or don't quite understand yet. And so, yeah, so that's, I guess, the answer that I would give to this question.
Great. Maybe Deepa, you want to look at the questions in the Q&A and answer the ones that you feel are most relevant, that you haven't already covered.
Okay, so one question, I guess, which is important, and I should have mentioned this, is how do we culture obligate symbionts in the laboratory in order to characterize them and perform studies on them? And thanks for this question. We actually can't in most cases culture them, right? Because by definition, in some sense, they are obligate, they can't survive, these bacteria can't survive outside the host. So people have tried hard to sort of build artificial systems where they can be able to produce these kinds of organisms in the lab and study them. But a lot of the study is focused on using microscopy to make sure that these organisms can be seen and they're marked in some way, and then they can be studied better without culturing them necessarily. In some specific cases, there are instances where you can culture some of them for at least some period of time, but there are problems there because then their behavior may change, right? Because it's not in the body of the organism that it normally is. So that is a challenge, and that's part of the reason why it's very difficult to do these kinds of studies. Okay, maybe one last question quickly. Can we yeah, so this is another very interesting question about cheating, right? So when you have multiple partners that are in a symbiosis, is there a possibility of exploitation of one partner by the other? And yes, absolutely. And there's a lot of very, very nice work that people have been doing on this possibility of cheating in a symbiosis. And it does happen. And there are counter strategies that the host or the other partner may evolve to be able to deal with the cheaters and make sure that cheaters don't take all of the benefit that is supposed to come to them. And there's a lot of policing, which is one possibility. Another possibility is cheaters pay a cost, so everybody else could gain from the cooperation in a way that the cheaters cannot gain. And so, that's a whole different field of evolutionary biology that a lot of people have done very, very interesting work on. In the context of these specific kinds of symbioses that I mentioned today, there are only some cases where cheating has been investigated and shown to occur, and we understand how it is controlled. But in these systems, we don't actually understand very well how cheaters are controlled or how they're dealt with. So yeah, so I think maybe that will be the last question. I'm sorry I couldn't answer all your questions, but maybe we can have another one of these sessions later.
Yes, thank you so much for answering all these questions, Deepa, and for this very interesting talk on symbiosis. I'd just quickly like to also thank Professor Sadamani for ideating this ecology series. And Professor Sadamani, would you like to say something about the series and our next talk as well?
Yes, I'm very happy to have Deepa give this very interesting talk. And it is evidenced by the number of questions. As many as 20 questions have come. And in addition, also we learned a lot about plant lichen interactions, insect bacterial interactions, and there are some cool ideas that were shared, such as how we can negotiate with a parasite to become good. So all this was very, very useful. And I'm happy that the students are showing a lot of interest, and I hope they will benefit from this whole series, ecology series. And thanks once again, Deepa, and all and the communication teams for organizing this. And next week, we hope to have Dr. Jayashree Ratnam, who's coordinating and helping with the MSc Wildlife Biology at our campus. And I am hoping that she will tell us a lot of little stories about how their MSc students have been learning on the wildlife biology all over the country. With this, I would like to thank the team once again. Bye.
Thank you. Bye.
Thank you. Bye-bye. I'm closing the meeting.
Mm-hmm.